Assalamualaikum and a very good day to everybody. So for group 2, we will be presenting about asexual reproduction strategies. So this is all of our team members, which consists of 4 people, and the work have been equally divided and everyone has contributed equally to the project. Next. So when we talk about reproduction in plants, we know that the topic is quite big and there's a wide range of strategies of reproduction to cover. So to make things easier, we are specified down into the topic of tissue culture. And to get more information about this field, we have conducted an interview with an expert on this part. About our interviewee, we have interviewed Dr. Muhammad Firdaus Ismail, and he is an expert in the field of agricultural botany. And this will help us to understand more and get more knowledge about our specific topic, which is tissue culture and reproduction.
Assalamualaikum warahmatullahi wabarakatuh. My name is AMIRAH WAJIHAH binti KAMARUDDIN. So today I'm going to talk about a method of a tissue culture. Okay, so as we know, tissue culture can be used in both plant and animal. Okay, but today I'm just going to mention about a tissue culture in plant. Okay, so without further ado, let's get started. Okay, so as we know, tissue culture is a technique in which a small fragment of a plant or its plant are introduced into an artificial nutrient medium which allow it to functioning or growth. Okay, so it is important to take note that the entire organism also used for a tissue culture, not only a small fragment of the plant. Okay, so the medium that can use for a tissue culture is a broth or agar. Okay, so agar is a nutrient dense gelling agent. Okay, so we know that plant tissue culture systems are effective used in a commercial production as it can produce in a large scale. So there's a few types of a tissue culture. That is, first is a seed culture, embryo culture, and an organ culture. Okay, so for seed culture, orchid is commonly used for this type of a tissue culture. Okay, so for this method, eggs plant or tissue from the plant are obtained for an in vitro derived plant and introduced to an artificial environment where they can get a proliferate or increase in number. Okay, so plant material that will undergo the method need to be sterilized first to prevent the tissue damage and ensure optimal regeneration. Okay, so for the organ culture is a type of a tissue culture that involves isolating an organ for an in vitro growth. Okay, so an organ plant that can be used is a shoot, root, leaf, and a flower. Okay, so the last one is the step of a plant tissue culture. Okay, for the stage 1, the tissue need to be sterilized first to prevent any microorganism that will negatively affecting the process. So in this step, tissue will initiate to culture. Okay, and the second stage, that is the in vitro plant material is redivided and then introduced into the medium. Okay, so this medium is composed of a component for growth that include a nutrient. Okay, at the stage 3, the roots are formed and the hormones are required in order to induce rooting and consequently complete a platelet. That is all from me. Thank you.
Assalamualaikum and hi everyone. So I am MAZLEENA. I will continue with application of tissue culture. So firstly is rapid clonal propagation. A clone is a group of individuals or cells derived from a single parent's individual or cell through asexual reproduction. Therefore, all platelets regenerated from a callus or suspension culture generally have the same genotype and constitute a clone. So, second is somaclonal variation. Somaclonal variation is like a genetic variation. So basically this variation has been used to develop several useful varieties. Third is transgenic plants. So, a gene that is transferred into an organism by genetic engineering is known as transgene, but an organism that contains and expresses a transgene is called as transgenic organism. The transgenes can be introduced into individual plant cells. The platelet can be regenerated from these cells. By this method, the platelet give rise to the highly valuable transgenic plants. 4. Induction and selection of mutation. So this is the flow. So firstly, mutation induction. Mutation induction is when the mutation are added to single cell liquid cultures. So next, it will isolation of induced mutation, where the useful mutants are selected for future breeding. So basically, it will test the mutation by tolerance it to stress like pollutants, toxins, salts, etc. Lastly, agronomic evaluation of mutants. The surviving healthy cells are taken to solid medium for raising resistance. So, lastly is the application by resistance to weedicides. So, firstly, the weedicides are added to culture initially in very small concentration. So, next, the dosage is increased in subsequent cultures until the desired level of resistance is obtained. Then, it will regenerate to form platelets and plants. That's all from me. Thank you.
Assalamualaikum and a very good afternoon to everybody. One group, and now we'll be interviewing Dr. Muhammad Firdaus Ismail from Faculty of Agricultural Botany. So first of all, I would like to thank you very much, Dr. Muhammad Firdaus, for joining us today. And we will be asking you a couple of questions, and it will be nice if you can answer it. All right, so Salam.
Thank you. All right, please go ahead.
All right, so question 1. What are the benefits of tissue culture?
The benefit of tissue culture. Well, tissue culture is the technology used in propagation for plants, okay, to multiply and produce plantlets, okay, in a huge number and in a faster time and in a more uniform way compared to other techniques. The benefits of it is that you can produce plantlets, okay, that is uniform. Uniform meaning that there is not much variation between the plantlets that you planted. It is beneficial if you are going to have a big monoculture system. Let's say you have thousands of hectares of land. All right, so it's easier for you to manage if the plants grow at the same rate, and also if they can produce fruits or any other yield at the same time. So tissue culture can give you this because they will produce uniform plantlets. And secondly is that plant tissue culture will allow you to produce mass number of plantlets, okay, at a time, compared to conventional propagation methods, okay, where it take much more resources, okay, and also much more time, since tissue culture normally could produce faster plantlets compared to other conventional methods, okay. But of course, it's depending on the species. At the same time, tissue culture is beneficial, okay, in terms of producing plants that is having scarcity in raw material or propagation materials. Okay, let's say that from one tissues, meaning that you do not have to have a lot of source, okay, for you to produce plantlets. And I think that's among the benefits of tissue culture.
Thank you very much, Doctor. So for our next question, we know what the benefits are. Now, what are the problems faced during handling out tissue culture?
Well, in handling out tissue culture, there might be several challenges or issues. The first thing is that in establishing or in coming out with the protocol itself, Okay, not all plant species, okay, will have a same protocol to produce that, meaning that we need to optimize the methods and protocol before it could be applied to different species of plants. And secondly, of course, the biggest challenge for tissue culture activities is contamination. All right, tissue culture can easily be contaminated in the process. Okay, so it requires a highly skilled practitioner or expert in handling or in producing tissue culture plantlets. And thirdly is that, okay, even though tissue culture is like a cloning technology, you can produce, okay, identical plants from the mother plants or identical plantlets with each other, but at the same time, okay, we need to be very careful because sometimes the plantlet or the plants that you produce tissue culture might not be, might do not express the real potential of the mother plant. There are cases where plants produced from tissue culture do not give the same yield as the mother plant, meaning that, okay, we need to do before tissue culture plantlet could be planted. It's good to evaluate them first, okay, to ensure that the plantlet that produced from tissue culture truly is not having any defect or abnormalities and can actually express all the potentials, same as the mother plant or as what we are hoping for.
All right, thank you very much. So for our third question, besides tissue culture, what is the most common method used in asexual reproduction besides tissue culture?
Asexual production actually is a very big topic and it's a very big area on itself, and there's so many types of asexual reproduction. Okay, in fact, some species they already have a normal asexual reproduction system by themselves. Some species we know that can produce offspring through their leaf. Okay, at the same time, the most common in our industry in Malaysia may be grafting. Okay, grafting technique. All right. So, for example, our horticultural crop for some of the fruits trees, and normally we will use grafting. Sometimes people use mature grafting. All right, and now people are experimenting with a lot of technique in grafting technique. Okay, for example, some people believe that maybe if we could graft 3 plantlets together or 3 seedlings together, okay, and then maybe the plant will grow faster and produce a better yield. So grafting is one of the most common technique in agriculture. Aside from that, layering, a layering technique. Okay, sometimes if you want a fast fruiting plants or trees, so normally you can do layering. Okay, meaning that you can take a mature branch and then you can do layering, and then because when you do layering normally, okay, a mature branch can grow faster and produce yield faster. And layering also come in many different techniques and methods. Okay, so I think the most common aside from tissue culture or asexual in industry is grafting and layering. Okay, aside from, of course, for ornamental, maybe people use cuttings right away. Okay, you can use stem cutting. All right, maybe you can just use a certain species. People just prefer to use a stem cutting and dip it or treat it with rooting hormones so that they can rooting faster. And of course, sometimes people also, aside from cuttings, okay, sometimes in certain species of ornamental plant, you can also propagate new plants from the leaf. Okay, leaf cutting. All right, I think that that's some among the common techniques used.
All right, thank you. So for our last and fourth question, of all the methods that you have just cited and the other methods that you know, what is the economically and financially best method to do asexual reproduction?
Okay, thank you very much. I think when you mentioned the best in terms of economic, okay, in terms of practice, it depends on the species. It depends on the crops. In the end of the day, everything, all techniques or all technologies, it depends on your value and objective. What are you trying to achieve? All right. So first, okay, you need to know what's your goals. And secondly, of course, the feasibility of technology at your hands. You need to consider that. Okay, maybe it will save you cost or save you in a lot of other areas. Of course, currently in our industry, grafting is one of the most common technique. First, because it is comparably easier to do. Of course, you need the train. Easier meaning that you can do it anywhere. Okay, and almost everyone can learn how to do it. Okay, and secondly, grafting is that you will produce because and then it will fruiting earlier. Okay, for certain plants. All right. So meaning that maybe you need to think when you're choosing a method, choosing based on the specific of plant that can save you time and also cost. And of course, but in terms of mass production, tissue culture is leading in this because tissue culture can produce huge amount of plantlet, okay, in one time. But of course, tissue culture have its own hiccup, which is it require specific place, all right, an established laboratory or a suitable sterile environment where you can conduct the procedures. So tissue culture can produce mass plantlets. Of course, it's a higher cost, but the economic return is actually quite good if you really plan use the right crop, okay, for that technique. So I think for conventional plants in Malaysia, you can use grafting, and then but for big plantation, you have the skill and the financial strength that they can opt for tissue culture. Okay, I think that's my.
All right, thank you very much, Doctor. So that's it for now. Thank you for sparing some of your time to answer some of our questions. And before we end, can we take a quick picture?
Yeah, sure.
All right, 1, 2. All right, thank you very much, Doctor.
Thank you very much. Okay, good luck and all the best.
All right.
Assalamualaikum warahmatullahi wabarakatuh. My name is MIQDAD. So today I will talk about conclusion that we get from topic and from interview. So without further ado, let's go.
For the first one is conclusion from the topic that we studied. Tissue culture is a plant propagation done in a laboratory under sterile condition. Tissue culture is line tissue cloning where it copies the same exact trait as the original plant. It can produce a plant with a large number, uniform and easy to grow. Aside from the advantage, since the tissue cloning copies the same trait, it will propagate pathogens too. So, if the parent plant is not bearable with some diseases and microorganizing the offspring plants, even this technique has some advantage and disadvantage, it is still an important technique to propagating many fruits and plants. Not only that, in educational part, many botanical garden and university use tissue culture for plant conservation. The second one is a conclusion from the interview that we do. The benefit of tissue culture are by using these techniques, it can produce uniform offspring plants and can produce mass number of crops. Comparing to the conventional propagation, it can also reduce the amount of resources to produce of offspring plants. There might also several challenges which are protocol to do the tissue culture, contamination of tissue culture during the process, and some plantlets do not express real potential as same as parents' plant. Besides, the other techniques used in Malaysia identify are layering, crusting, and stem cutting. Now, crusting is the main common technique since it is easier to do, but in terms of mass production, tissue culture none the first that place right now. So that's all from me. Thanks for watching. Thanks for your attention. Have a nice day and Assalamualaikum.